Good morning, good afternoon, or good evening, depending upon where you are in the world, and welcome to today's MDDI webcast, Urgent Update on Changing ISO Standards, Staying on Top of the Regulations, sponsored by Nelson Labs and broadcast by Informa. I'm Chris Keach, and I'll be your moderator today. We have just a few brief announcements before we begin. First, the slides will advance automatically throughout the event, and this webinar is designed to be interactive. The dock of widgets at the bottom of your screen will allow you to learn about today's speakers, download resources, share this webinar via social media outlets, and participate in our Q&A session that takes place at the end of our presentation. Please note that we'll try to get to as many of your questions as we can at the end of today's program. However, if we're unable to get to your individual question today, someone will get back to you after the program is over. And again, the slides and video will advance automatically throughout the event. You may also download a copy of the slides via the Resources widget. Towards the end of the webinar, we will ask you to complete our survey found on the right-hand side of your screen. Please take a moment to fill this out before leaving us today, as your feedback will provide us with valuable information on how we can improve future events. And lastly, if you're experiencing any technical problems, please click on the Help widget found at the bottom of your screen, or you can type your issue into the Q&A text area, and we will be glad to offer one-on-one assistance. And now, on to the presentation. Cytotoxic Reactivity, Understanding the Cause and the Path Forward. Discussing today's topic is Helen Ragel, PhD, biocompatibility expert at Nelson Labs. To learn more about our speaker, please visit the Bios widget. It's with great pleasure I now turn this special session over to Helen to begin. Helen, take it away.
Thank you, Chris, for the introduction. I thought that before we start, I would want to give you a little fun fact about me, about like a personal fun fact to share with you today. So I don't know if many of you know this, but I actually was born and raised in Estonia, this tiny little country in northeastern Europe. And it's so tiny that when my husband and I met and I said, oh, hi, I'm from Estonia, he was like, that doesn't exist. So of course I had to pull up the map and prove him that it's actually there. And then so he looked at it for a second and he was like, that's Narnia. And I just wish that traveling there would be as easy as that, you know, just through the wardrobe. So that's the fun fact about me. Before we go into today's webinar and really dive in, I wanted to share another story with you. This is a story that happened to me just a little while back at work. So what happened was, I was sitting at my desk and I was having a conversation with the sponsor on the call. And then suddenly, we were talking about my favorite subject, biocompatibility. And then suddenly it was like, the fire alarm came on. And I was thinking, okay, should I just ignore it? You know, I'm in the middle of something. 99% of the time, it's going to be a false alarm anyway, so should I go or should I not? You know, should I stay or should I go? But I decided, okay, since, I need to follow protocol, I got up and I looked for the nearest exit, right? Even though usually I take the stairs a little bit further down the corridor. But I was like, no, in case of an emergency, you have to use the closest exit that is the most applicable. So I did. After, sitting, under the burning sun of Salt Lake City for 20 minutes in a parking lot, of course it turned out to be a false alarm. But while I was sitting there in the parking lot, being fried by the beautiful August sun of the desert, I realized this is what also happens to us when we talk about cytotoxicity and cytotoxic failures. Because trust me, when we talk to clients, a lot of the times they run into this problem. And so I thought this story is kind of like a good analogy to give to help you understand this is not something new that we're facing. This happens quite often. So I want you, next time when you run into this issue, have this alarm go in your head, off in your head, and think of me and this presentation, and I give you bonus points for sound effects. Okay, so in today's webinar, we'll go over the cytotoxicity test, why we do it, what is it, and what does it really tell us about the medical device. We will also discuss some known culprits that are, we've seen that can cause reactivity in this cell system, and we will talk about how to interpret the data and what to do next, right? As the cytotoxicity failures are happening quite often, as I mentioned. I mean, really, I would say maybe even weekly we have these discussions with our sponsors. We also published recently a review paper in collaboration with AMI Journal to discuss all of these things, what can be causing the failures, and how to proceed from there. So I encourage you to look this up, and I hope that you find it useful in your investigations. So we discussed using the alarm as the analogy or the notification system when you have a cytotoxic response. And always when you have an alarm, it's good to have a person by your side who knows what to do, like a safety officer. So for today, I will be your safety officer, and I will help you think through different options that can be applicable to your situation, and then when you do run into this issue, then you will know what to do. Okay, as a safety officer, I have to have an evacuation plan, or in this case, an exit plan from the concern to define, okay, what should we do, where should we go, and what's next. But in order to know what you really need to do and what is the best route of exit, you also need to know where you stand within the framework of that plan, right? And based on that, you need to define whether the best exit route is through here, or would you need to exit through here, or maybe this is the exit that is best for you. So later in this webinar, we will talk about the different exit routes and how you can use the best one for your specific situation. But before we go there, we need to get back to the basics. So why? Why do we even do cytotoxicity testing in the first place? I mean, if we know that it's going to have, potential failures, and this is really the test that we've seen to have the most failures out of all the biocompatibility testing, then why do we even put ourselves through it? Every medical device that has contact to the patient or the medical personnel has to go through a biocompatibility assessment to make sure that it's safe to be used. A biocompatibility assessment is performed per ISO 10993-1, which is the framework to do this assessment. And as you can imagine, the specific risks associated with the given medical device are tightly tied to its use, its intended type of contact and duration of contact. Now, table A1 in ISO 10993-1 categorizes all the medical devices out there into distinct groups based on their contact type and duration. And based on that, defines the biological endpoints that should be addressed, either through risk assessment and justification or through testing. And as you can see from the table A1, regardless of the intended contact, the endpoint of cytotoxicity is required to be evaluated for all medical devices that have any contact to the patient or the medical personnel. So that's really why we have to do it. It's basically required. And since the cytotoxicity test is a fairly quick and simple and inexpensive test, in the realm of all the biocompatibility testing out there, it is often the one that is actually performed rather than using a rationale or justification for not doing it, right? And also, it is often used as an initial screening test to see if there's anything concerning present in the device extract. So that's why we have to do it. Okay, so it can be either seen as a blessing or a curse, and of course, sometimes it could be a curse. Since it's a highly sensitive test, it has many positive attributes, but you can see that the supersensitivity of this test can also lead to many drawbacks. Now, the plus side, we typically use this test to perform the initial screening like I mentioned. For example, if you have a new medical device and we want to start testing it, it's really good to run the in vitro systems first to make sure that we're not, potentially exposing the test animals to anything potentially hazardous. And this is also something that ISO 10993-1 says, that we should really do the in vitro test first and then move on to the in vivo methods. Also, we use this test often in the medical industry, medical device industry, when we have a verification step. So, for example, when we have an already marketed device that is undergoing a change, for example, and we're changing slightly the configuration or maybe the materials, proportions within the device, then with the cytotoxicity test, if the changes are minimal and the test demonstrates comparable and good results, then additional animal testing, which is both time and cost-consuming, might not be needed, given that you have a good justification for not pursuing this other testing. Also, I have to mention that the world of research uses this cytotoxicity test a lot as well. And because of that, there's a lot of information available in published literature about all sorts of materials and their reactivity in this cell system. So that's good. There's lots of information out there about how some materials might react in the let's say, a cytotoxicity test. So as I mentioned, on the other hand, because it is an in vitro test system where you have a single monolayer of cells just hanging out there in the well, and you put the extract on there, any slight disturbance to the environmental conditions such as osmolarity or pH could have an impact already and cause the cytotoxic response. I always say to my clients, we have to take a step back and think, if you add, let's say, a couple of drops of pure water into the well with the cells, you're most likely going to see the cells die. And, we know that the water in general is not toxic because I'm, drinking it right now, and we'll hopefully see that I'm not going to have any adverse reaction. But of course, we need to understand what is happening when the cytotoxic reactivity is seen. And we need to make sure that if we see the failing score in the cytotoxicity test, that this is not something that could potentially have an impact to the patient. And we'll talk about this a little bit later on. Okay, now, the go-to document to cytotoxicity is ISO 10993-5. It gives guidance on various test methods that can be used for cytotoxicity testing. It also provides the test overviews and setup, as well as the acceptance criteria, which is good. Because, as I mentioned, the cytotoxicity test is often used in research, but the issue is that between labs, the way that they do the test can be very, very different. That makes it kind of hard to compare the data. So in that regard, the ISO 10993-5 really provides the set framework to perform the testing in a way that is standardized throughout the medical industry. So kudos to ISO 10993-5. So as I mentioned, the ISO 10993-5 provides various methods for cytotoxicity testing, and roughly those tests can be grouped into two groups, qualitative tests and quantitative tests. Historically, the medical industry has been using MEM elution assay for the assessment of cytotoxicity. And the MEM elution assay is a qualitative assay where the impact of the device extract to the treated cells is visually observed, and the cell density and morphology and overall appearance is reported. For this assay, the person performing the observation has to be properly trained to be able to adequately evaluate the results, right? And based on the observations that that trained person does, a score is given to the test article cells, but also the negative and positive controls. So the key benefit here is that you have this visual feedback, and you can really identify and see potential changes to, for example, cellular characteristics, for example, like cell shape or intracellular structures, or something that could indicate that something is going on, which might be good supplemental data to have. The downside, however, is that because there is a human element, there could also potentially be some bias introduced to the results. The scoring table is also provided in the ISO 10993-5, and according to the standard, a score of 2, that's where the dashed line is, or less, is considered as passing. Now, I do have to point out that we have seen some regulatory bodies also want investigations to a score of 2, because it's kind of teetering there at the threshold line. So we have seen that happen, so just be aware. And also, you should also kind of think about it, okay, if you're using the cytotoxicity test as a screening assay or like a verification step in your quality system, and you've always gotten like the 0, and then now all of a sudden you get a 2, obviously that is something that should be also further investigated to understand what has changed in the process and what impact that might have to the final finished product. Now, quantitative assays, those tests use the uptake and metabolic activity of live cells to detect their viability. The typical tests that are run under this category are MTT assay and XCT assay, which use a specific dye in the solution that is taken up by the cells and metabolized to this beautifully purple color, which can then be measured using a spectrophotometer. There are other quantitative tests also listed in ISO 10993-5, for example, neutral red uptake, but they still use the same method, where there's an uptake to cells, and then we measure that based on, that the fact that only the live cells can do this. These tests are good, these quantitative tests are good because they remove the bias of the observer, right? So there's no human element. And it gives a percent value for cell viability. And this percent gives you viability from 0% to 100%, which is a bit wider than the scoring system that is used for the classic MEM assay, where you have a score from 0 to 4. So that's good. However, it does not provide any additional data on the morphology of the cells, So there's no visual input, other than the pretty colors that we see on the plate. Okay, so according to the standard, reduction of cell viability by more than 30% is considered a cytotoxic effect. I am more like a glass half full kind of person. So in my mind, anything 70% or higher is considered a passing non-cytotoxic reaction. So what happens if we see a failure in the cytotoxicity test? As you hear that sound in your head. Let's use the approach here of using the evacuation plan. So we need to know the plan, okay? We need to know your situation. And as I mentioned before, the situation in your case and the specific exit that is most applicable to your situation really depends on what is your device and what available information do we have on the device. We'll go through a couple of examples of exit plans in just a few moments, but I first wanted to emphasize again that we cannot ignore the alarm, right? Even though we are like, oh, again, it could happen, and we know that a lot of the times it's probably a false alarm. Regardless of that, we need to understand first, is it coming from the device? Is it something concerning? Or we need to demonstrate that actually it's something in the test system, the way we run the test, that could be resulting in this reactivity. So we know that some materials, for example, are not compatible with the in vitro cell culture system. For example, we had this one sponsor that came to us and they said, okay, we have a cytotoxicity test that we use regularly in the screening process of a quality control step. And they saw zeros, like I mentioned before, all zeros, and then suddenly there's a 2. And so what is going on? How can we fix it? And we looked through their manufacturing process, and nothing really seemed to pop out. But then when they went to visit the facility where they were making these devices, they noticed that actually they had implemented a little change in the process. And all the people that were handling the medical device were now wearing gloves. Can you guess which gloves? They were latex gloves. And latex is known to cause reactivity in this particular test system and is therefore often also used as the positive control in the test system. But knowing what we know about latex gloves and the fact that they are quite widely used in a clinical setting or academia, we know that the actual concerns from patient safety perspective are not are low. So there, in this case, we could prove that actually it was a false alarm. Okay, so let's talk about some known culprits that we know that could result in elevated reactivity in this test system. We know, for example, from our own experience as well as published literature, that some metal ions, for example, silver or copper, might present a cytotoxic reaction. And also some plastics, especially softer plastics that may contain plasticizers that could be released during the extraction, those could cause a failing result. ISO 10993-12, the one that talks about the sample preparation, also emphasizes that some materials like elastics could result in reactivity. And even in that standard, it has said that for those specific materials, a lower than standard extraction ratio might be used. The standard is the 3 centimeters squared per milliliter, and they say you can potentially use, if you have a justification, a lower extraction ratio because it's known to cause reactivity. Cytotoxicity test is also complicated for devices that are highly absorbent or that may start to, let's say, disintegrate when they're fully immersed. Like, for example, we see lots of failures with electrodes, right? but that's because, you put the electrode onto the skin, but in a clinical setting, but we cannot really do that in the test. So a lot of the times it's fully immersed into the extraction medium. And there you can see how the electrode hydrogel material starts to disintegrate and start to really alter the environment of the extracts, and that could be something that is causing the reaction, not that the electrode itself has any potential to have an impact, adverse effect in the patient. So we have to realize that as medical devices are such a diverse group of products, we cannot highlight all possible culprits here, right? Because that would just be, see you in 10 years. But why we're having that is why we need to know all the background information about your specific product when we're seeing this reactivity. And there you can really see that this background knowledge will give you an advantage to identifying the potential source or sources that could be resulting in this reactivity. The more you know, the better you are off. So what do we do? What do we do when we have a failing result? Where do we go from here? So when we hear the alarm, we need to clearly take a note. We need to investigate. We cannot ignore. We want to know the root cause. What is causing this reactivity? So ideally, what we want to find out is that if we have a medical device that contains lots of different components, can we identify the component that has reactivity? Because then you kind of heard the question into more specifics. And if we know that, okay, this component has this reactivity, everything else is good, then we can look at what we know about this specific component and if there's any data available to really understand what is it and if it's going to be an issue for the patient. Also, that would help us identify whether we need additional testing or what additional testing would be most beneficial to pursue this investigation and determine the actual risk to the patient. Now, it really depends on what information is available, right, or can be generated to mitigate the concerns from the seed reactivity. And the main question we want to answer here is, if this device is going to be used on a patient, are there any actual risks associated with the intended exposure? That's the real question that we want to answer. So let's discuss some specific examples of what we can do in order to find what are the risks. Okay, so our first example is silver-containing wound products. I hear some of you like, oh, yes, because the wound products in general are pretty hard to test, and silver-containing even harder. So we know that silver is typically added as an antimicrobial agent to some of these products, right? And we also know that silver might react with the cells in this in vitro system. So the key really is to understand how much silver is going to be exposed to the patient, right? So this can be either identified through first available information. For example, if you know exactly how much will be placed onto your product, and then you can use that worst case amount for analysis. Or alternatively, you can also address this via additional testing by looking specifically at the silver that may leach out of your device during use. So now, the second step is then to define what are the permissible levels to the patient using guidance from documents such as ICH Q3D, right? And these are specific guidance documents that look at potentially allowable levels for metals, okay? So this most recent revision, the R2 of this document, ICH Q3D, indicates that for oral exposure, for instance, the allowable daily limit for silver is on 167 micrograms per day, each day. Oral, we use oral as, worst case, because when we have, let's say, something that is on the skin, the oral exposure is, considered worst case, so we can use that level. If you have, of course, skin exposure, topical exposure levels, you could absolutely use those as more relevant. But in this case, if we can prove that the amount of silver present at total amounts or that is able to leach out of the device and be exposed to the patient is below that permissible level, then the toxicological concerns are considered to be low, right? And the risk from this reaction in the cytotoxicity test in general to the patient is considered low. Okay, another example of metals, and we love metals, right? We had nitinol. Let's say use nitinol. We know nitinol is very widely used for implantable devices because it's high strength, it's corrosion resistant, and overall it has been shown to have a good biocompatibility profile, right? But sometimes, even with metal devices, we do see that, that there is a cytotoxicity failure. And especially we see when they are very small, like the staples here. So we had this example in-house where we have nitinol staples, and they're very small. So for cytotoxicity tests that requires you to use a specific extraction ratio per the standard, we have to pull like 1 million of them together to get the required amount for testing, the required amount of extract for testing. So in reality, however, we're not going to use 1 million of them on the patient because then you will turn into Robocop. But what we need to find out is that we need to understand that if something is really small, the way that the test is run or the test setup might not reflect clinical situation, right? And if something is small, then it's really likely that that is the case. So always make sure that you take into consideration the way that the test was run. So in this case, we would typically recommend to have some additional data available to supplement the potential concerns from patient perspective, right? And we have to always acknowledge that the depth of additional necessary data is, of course, dependent on the context duration, right? In this case, because we have implantable devices and they will be there forever, I would recommend looking at metal release over time. For example, nitinol has nickel that might have leached out and caused this cytotoxic reaction when we have 1 million of them together in one pot. So what we want to know is that from a toxicity perspective, is there a concern? So what we could do, we could pull the amount that is the worst case amount and run chemistry for metals, maybe specifically nickel, to show that the nickel in this case, in a clinical use scenario, is not going to have any concern. But you can also use, for implantable devices, especially metal devices, usually we recommend to have supplementary data anyway using chemistry, like through extractable leachables testing, so you can use that data to supplement this question as well. So in this case, we were also able to confirm that the allowable levels that are allowed by the standards or the guidance documents and the amount that was coming out of these nitinol staples was, the amount was much lower than the standard says is okay, and we were able to prove again that in this case, the risk of the nickel release in clinical use scenario was low. At the moment, I do have to say that numerous metal implants, especially implants made of cobalt chromium alloys, are under very strict scrutiny. And I would say that similar investigational approach may be followed for these devices if you see a cytotoxic response in your devices. But of course, we have to kind of take a step back and think, okay, implants that are like hip implants, for example, when there is load-bearing needs to them, and they may experience wear during use. In these cases, the amount of potential metal release could be much higher than just like a standard sitting device that is being extracted and assessed. So for these types of scenarios, you might have to supplement this additional data as well using wear testing and metal release from simulated use condition scenarios. But I have to say, this is another story for another time. Okay, our last example today touches the contact type aspect. We sometimes see that cytotoxic reaction is happening when we immerse the soft plastic containers or tubing into the cell culture medium during extraction. I do have to mention that typically in ISO 10993 based testing, they use both a polar and a non-polar extraction separately. But for the cytotoxicity test, this one is special. Very special. They use one single solvent, this is the cell culture medium supplemented with serum, and because it has serum and medium, it contains both aspects of polar and non-polar characteristics and is often referred to a mixed polarity solvent. So, in general, if there is something that is, let's say, non-polar that might be extracted out in a non-polar solvent from these tubings, that would also be captured in this test, right? But we know that sometimes these tubings and containers that, let's say, hold IV solutions or are only meant for specific applications like irrigation, where they will only see a polar solvent, the device might never contact a non-polar solvent at all, right? So really, is it really relevant for analysis? I would say the short answer is no. And I would say also that ISO 10993-12, the one about sample prep, also agrees with me, because it says that in cases where we know that there's only one type of contact, then that one extraction vehicle might be used, right? And for cytotoxicity tests, for example, it says that polar extraction vehicle like culture medium without serum might be used. So I would then think, would that make sense to retest the cytotoxicity test with the clinically relevant solvent type? So you have to kind of take that into consideration as well. Okay, these have been a few of the examples where we have worked through the problem and found a good exit route for the sponsor. Of course, as I mentioned earlier, due to the vast area of different medical devices out there, one quick, easy solution cannot sadly be given. If I had the magic 8-ball, I would totally give it to you, but sadly it's not, I don't have it. But I would say that in general, this investigational approach, this step-by-step evaluation strategy, has worked quite well in the past. So, to recap, when you see a cytotoxicity result that is saying, oh, you have a failure, please do not ignore it. We need to understand where it's coming from and whether it's going to be hazardous for the patient. So we need to investigate where is it coming from, and we need to identify the potential source and understand whether it has a potential impact to the patient when the medical device is being used in the way that it's intended to be used. And, I mean, this is what we're really here for, right? We want to ensure that the medical devices that are being developed for the patients are good and they do no harm, as the Hippocrates once said. So with that, I wanted to thank you for having me as your safety officer. It's been awesome to talk to you about this because, biocompatibility is, my favorite. And I do have to say that our mission here is to help you and other medical device manufacturers demonstrate the safety of your products, right? And through that, we want to safeguard the global health. So I'm really happy to be in your task force. So Chris, over to you.
All right, well, thank you, Helen, for a great presentation. We're going to now move on to the question and answer portion of our event. As a reminder, if you'd like to participate in our Q&A session, simply type your question into the text box located to the right of the presentation window, or you can click on the Q&A icon at the bottom of your screen, type your question in, and then click the Submit button. Please note that we'll try to get to as many of your questions as we can here in the time that we have left. However, if we're unable to get to your individual question today, someone will get back to you after the program is over. So, Helen, we've got quite a few questions coming in. Our first one for you, what are your thoughts on evaluating end-of-device life cycles on aged samples just by a cytotoxicity test?
Yes, so that's a great question because that's something that with the, submission to the NDR, for example, has now become really like a key question, right? And I do understand that we don't want to have everybody undergo all of the testing again to demonstrate that, yes, your device at the end of shelf life is still biocompatible. So what we have, developed as a strategy here is to use the highly sensitive nature of the cytotoxicity test as an initial screen, and then compare the at the end of shelf life versus the newly manufactured device. And if they show that they're really comparable, you visually cannot see anything different, then I would say that's a good place to start. Of course, if you do see some reactivity, that needs to be further investigated, and then you can pull us in to help you through that process too. But I would say that the initial strategy of using the cytotoxicity testing for that makes really good sense. Of course, it depends on if you have devices that have blood contact. There's other concerns there too, but. Overall, the cytotoxicity test for end of life, I would say is a good approach.
All right, thank you. And we're going to move on to another question here for you. Our next question asks, can clinical in-house history data be used to justify cytotoxicity?
Yes. So we always say that if you have something that is already out there, right, and you see that there is a cytotoxic reaction, let's say, again, these wound products, right, we do see lots of times that there is an issue with the cytotoxicity test. So really what I really wanted to show with this webinar is that we need to answer, is this reaction in the in vitro test system going to be also impactful for the patient, right? And if it's something that we can show, like for example, topical products, it's easier because you can see the reaction, right? Is there, irritation potential or sensitization potential? Is there, I always say like the closest to cytotoxic reaction would be, necrosis. If you don't really see that and you can prove that with your clinical data, I would say that clinical data is very useful in arguing that your cytotoxicity test might not give you the accurate result, right? And it might be the false alarm in this case. But you do need to have some data to prove it, that it's not going to be harmful for the patient.
All right, thank you, Helen. And as a reminder to our audience, if you'd like to ask a question, just go ahead and click on the Q&A icon, type your question in, and then click the Submit button. We'll get to as many of your questions here as we can in the time that we have left. And again, if we don't get to your question today, someone will be getting back to you after the program is over. So, Helen, our next question for you, can you recommend, or can you comment, I should say, on cases where we see a higher toxicity, such as grade 2 at 24 hours, and that decreases to 0 at 48 hours or 72 hours.
Mm-hmm. Yeah, that's a very interesting case, right? Because normally you would expect that if you see something already at 24 hours, it will still remain there after 72 hours, right? But we have seen sometimes that happen, especially if we have some medical devices that have a lot of volatile organic compounds being extracted in the extraction process, and those are, they could be there at the initial time point, but after a while they will volatilize away. So then their presence would reduce in the extract. So that's one example where this could be. But again, additional data might be needed to have to supplement, like, where could it be coming from, and to really prove that the amounts that are present, they're okay to the patient.
All right, thank you, Helen. And we'll move on to another question. This one asks, when can known data for any product be considered sufficient to rule out the need for cytotoxicity testing?
Yeah, so that's a tricky question, because I know, again, we can always say, like, the cytotoxicity test is so flimsy. Why do we even do it if we have such good, like, clinical data or whatever, right? But I do think that it is valuable in having that data, first of all, as a verification for, let's say, regulatory bodies, like, okay, we did do this, and it's really not going to have an impact. And I mean, there's really, if you have like a silicone, most likely you're not going to see a cytotoxic response. And since we want to use this data to show that really the concerns are low, I think it would be still good to run through it, especially now with the MDR, where we really want to, show that at the first point of coming out of the facility, of the factory, and at the end of life cycle, there's no reaction happening. It's good to have those 2 points in parallel with the cytotoxicity test to show that there's really nothing happening over the course of the shelf life.
All right, thank you, Helen. And our next question asks, do you think we can justify a failure with in vivo data? Yes.
So I do always say that we need to look at all of the data, right? When we have a cytotoxicity failure, we need to look at all of the rest of the available data to see if there's something popping out, right? For example, we had this issue where there was some medical devices going through multiple rounds of reprocessing, and there was we have presence of, like, cleaning agents and whatnot that could result in the cytotoxic failure, right? And then we looked at, okay, we know because time 0 there was nothing, right? And now there is, so there must be something happening there, right, through the reprocessing. So we know what is being used, right? That's the specific detergent. And then we looked at that and we said, oh, okay, it could potentially have an irritation response that we might have caught with the cytotoxicity testing. Okay, so should we supplement that specific test to justify that, again, the risk to the patient when the device is being used on them is not going to have an adverse reaction in that in vivo system. But we also can use, lots of times we can also use implantation testing. Might also be really good to see how that implant really reacts in that specific tissue sample, right? If there's no reaction there, that might be also good information that you can provide to supplement that risk from the cytotoxic failure is not going to transfer to the patient.
All right, thank you, Helen. And we've got about, 7 minutes left here to answer some more questions. We're going to answer as many as we can. Again, if we don't get to your question today, someone will get back to you after the program is over. And Helen, our next one asks, while performing a risk assessment, What is the correct wording for harm due to cytotoxicity?
Yes. So, as I mentioned, like when we talk about cytotoxicity, it could be many things, right? In general, when I've talked about these things with the regulatory bodies, they kind of fall to the cytotoxicity, and the toxicity kind of indicates like systemic toxicity potential, right? And that's why a lot of the examples that I gave were about supplementing that side, right? But as I mentioned, we need to also kind of consider that toxicity itself is only one endpoint, right? And we need to consider other endpoints as well. So really it depends on what available information do you have in that specific scenario about your device and how it's processed to identify what could be the real concern. And also, like, for example, the cytotoxicity, right? I mentioned like a necrosis on the skin, right? I mean, that's, a little bit taking it further than maybe needed. But when we have something that is implanted, we can't really see that, right? And then we need to really supplement some additional data to prove that, it's not going to cause a systemic toxicity reaction, or if it's implanted in the brain, right? We need to make sure that doesn't transfer to some neurotoxic effects, because we when you have, especially like brain stuff, we don't want to have the cells die. So we really need to make sure that we're not ignoring the cytotoxic response, and we are proving with the appropriate data sets that reactivity is not going to be translated to the patient.
All right, thank you, Helen. Our next question asks you, how do you conclude if we see cytotoxicity in neat and 25% dilution, but not in 50 or 75% dilution?
Yeah, that's a great question, because, when we talk about cytotoxicity failures with regulatory bodies, they always fall back to, oh yeah, maybe you should do a dilution series. And I always say, like, well, what does the dilution series really tell me? But in this case, when we do recommend dilution series and we see like, okay, the neat is, cytotoxic, but then it's going to start to go away, which is, we expect to have that happen, right, with dilutions. We want to show it in comparison to something else, right? Then we can really say, like, look, this is our device, and it has, a cytotoxic reaction at neat concentration, but at 1 to 4 dilution is already okay. But as you can see, a comparator product that is already on the market shows cytotoxicity already still past that, right? And then you can kind of prove that your device is actually doing better in the cell system, and with supplemental data from other testing that you might have, you can prove that this cytotoxicity reaction is not really going to be a concern. Now, we do have to make sure that we understand what the dilutions do, right? Because yeah, it's a never-ending battle. I always say, like, okay, if we just do series. Another thing you want to consider is as well is that how your device is going to be used, right? As I mentioned, like, for example, with this electrodes and the hydrogels used on the electrodes, right? The way that the test is set up doesn't really mimic the clinical use scenario. So there is an additional justification or like a thought process that you could do to define, okay, the need when you have everything, extracted there, like worst of the worst case scenario, does that really mimic my clinical use scenario?
All right, thank you, Helen. We have time for one or two more. We'll see how long we have left after this one. Would you recommend to have the cytotox for each component that has contact with the patient, or should it be tested as a top-level build of the device?
Yes. So in general, what we want to do is we want to test first the final finished product, right? And really focus on the patient contacting parts, because those are the ones that matter, right, from the biocompatibility perspective. If you see that the final finished product does have some reactivity, then I would move into the component testing. Actually, if you do take like one component, and then you test them in a way that it really follows still that 3 centimeters squared per milliliter, or that specified extraction ratio that would be then, can be drawn back to the final finished device, then, literally it should be the same, but I think that regulatory bodies really would want to see first the final finished product being tested as a complete, to see, the overall reaction there.
All right, thank you, Helen. We're going to sneak one more question in here. Our final question for you. Do you recommend literature be used to support the cytotoxicity? And if so, do you have any specific databases that you can refer to?
Mm-hmm. Okay, so as I mentioned during my webinar, is that there are lots of data available, right? And if you have, let's say, a metal that you know is causing this reaction, you could find from the published literature an IC50 value that correlates to, let's say, the L929 cells that are used standardly in this cytotoxicity test. What we really want to know is that, again, what is coming out, what has been shown to be, cytotoxic, which is also presented in our paper for a lot of different metals, so you can go and look at there for valuable resources. And then you need to have a toxicologist look at the tolerable exposure versus what is coming out of your device to determine whether there is a potential impact or toxicological concern to the patient. So that's really what we want to do, is like we want to have what is coming out. If you have data to show like, okay, this amount is already shown to be having a IC50 that it will make a concerning result in a cytotoxicity test, and then prove with available data that from the toxicological perspective, this is not going to be a concern for the patient.
All right, well, thank you, Helen. That is all the time that we have for questions for today. Once again, if we did not get to your individual question, someone will get back to you after the program is over. I'd like to thank Helen, MDDI. Appreciate your time and your expertise on today's topic. We'd also like to thank our sponsor, which is Nelson Labs, as well as everyone in the audience. We appreciate your attention and your participation in today's event. Within the next 24 hours, you'll receive a personalized follow-up email with details and a link to today's presentation on demand. So please feel free to invite your colleagues and peers who may not have been available to listen to today's event. This webinar is copyright 2021 by Informa. The presentation materials are owned by or copyright by Nelson Labs and MDDI, and the individual speaker is solely responsible for her content and opinions. On behalf of our guest, Nelson Labs and MDDI, I'm Chris Keach. Once again, we'd like to thank you for joining us, and we hope you have a great day.